So let's kick it off. I'm Sammi. I am your host for this webinar. I am a licensed professional counselor associate. I'm an advocate and I'm a big member in the CF community. My personal and professional experiences have shaped a deep commitment to supporting individuals and families navigating chronic illness, disability, mental health challenges, and life transitions. So, as a sophomore in high school, I was diagnosed with a mycobacterial lung infection, the MAC, and I require intensive antibiotic treatment. I was getting daily IV therapy. And while the treatment was necessary to protect my lung health, it ultimately caused significant hearing loss. I've been actively involved with the Cystic Fibrosis Foundation, Cochlear Americas, the CF Research Institute, and advocating for greater awareness, accessibility, and more patient-centered care. Through these roles, I have gained a greater understanding of challenges of individuals and families, what they face during navigating complex healthcare and chronic illness. So these are just some pictures that I wanted to share with you all of my journey throughout CF care from multiple stages of my life. And here you're seeing pictures of when I had a lobectomy a couple of months ago, when I graduated from graduate school to get my license to be a therapist. There are some pictures of my mom, my best friend combing my hair after my lobectomy, some pictures of me doing presentations at counseling conferences about research and mental health with people with chronic illness, and some shots of me pre-lobectomy, some with my lung function, some shots of when I received an award from the Connecticut chapter, just a little Snapchat of my life with CF. And I want to also introduce you all to the panelists that you're going to be hearing from today. So here are your wonderful speakers. So we have Dr. David Nichols. He is the Senior Director of Clinical Research Development for the CF Foundation and an affiliate professor of pediatrics at the University of Washington School of Medicine. He co-leads efforts to develop new therapies addressing infections and other manifestations of CF while supporting academic research projects that help to establish best care practices and use of approved therapies. And then we have Dr. Stacy Martiniano is a pediatric pulmonologist and physician scientist whose research focuses on improving outcomes for individuals with CF. Her work centers on understanding and managing nontuberculous mycobacterial infections in people with CF, including identifying factors that influence susceptibility, diagnosis, and treatment response. Lastly, we have Dr. Rebekah Dedrick. She is a director of research at the Cystic Fibrosis Foundation, where she leads the academic anti-infective portfolio and manages strategic research alliances with industry partners to advance new therapies for people with cystic fibrosis. She's a trained microbiologist. Her research has focused on bacterial phage, I can never say that word, biology, and the development of transitional approaches to treat drug-resistant infections. So those are the wonderful people you're going to be hearing from. So without further ado, I'm going to hand it over to Dr. David Nichols to share a little bit about what he's here for.
Great. Thank you very much, Sammy. And it's really great to be with you all. I'm just going to pull up my slides for a moment. Okay. Thank you very much for taking an hour to be with us and to review some of the research that the foundation is supporting and what we're excited about here in the years ahead. I think we're all aware that over the last 10 years, the state of CF health and even including the state of lung infections has changed, and that mostly is due to the impact of modulator drugs such as Trikafta and others. And so we have now learned here many years into use that these modulator drugs, which aren't antibiotics per se, have had substantial impacts on infection in the lungs of people with CF who can take them and who seem to benefit well from them. We see reduced amounts of bacteria in the lungs of those starting these drugs by perhaps tenfold. Perhaps even more importantly, we see reductions in the percentage of people in the US who are growing Pseudomonas aeruginosa, for example, or Pseudomonas. Over 10 years in the registry, that number has decreased from about 50% of people in the US with CF down to about 25%. And so that's exciting, but it does create some new challenges, which we see as also new opportunities. The first one, I think many of you are aware of, even based on some of the questions some of you sent ahead of time, which is this. It's actually more difficult to monitor which bacteria are present in the lungs of folks with CF now. And that's because it's harder to provide sputum samples for culture, especially when you go into the clinic and you're asked to do that on command, so to speak. So we've seen a decline just from 2019 over about 4 year period in the number of sputum cultures that were sent by over half. And so there are efforts that are working on trying to make sputum collection easier for people when they come to clinic. We absolutely support those. But in parallel, we want to see if we might be able to get away from sputum to use a different source material to really know with the same amount of confidence what's infecting the lungs of people with CF. So we're exploring opportunities looking at breath and blood and urine, and I'll explain a few of those on the next slide. Somewhat related to this, we also see that the pool of people with CF who have these infections, let's say Pseudomonas, for example, who feel a need to develop new antibiotics or new antimicrobial treatments for themselves is somewhat smaller than it was in the past. And so that's good in some ways, but it doesn't negate the fact that this remains a key priority for us at the foundation because there are substantial numbers of people who continue to be challenged by these infections, which can lead to progression in their lung disease, but can lead to also challenges with exacerbations or symptoms on a day-to-day basis or trouble keeping up with a lot of medications and therapies. And so we need to find new ways to continue to successfully and feasibly develop new therapies. And one we're pretty excited about is called 1 bronchiectasis. Bronchiectasis is just a medical term we use to describe the kind of damage that happens in the airway when you have a persistent infection or inflammation in there. And it turns out CF is only about 10% of the broader population of people who have bronchiectasis. In other words, 90% of people with bronchiectasis don't have CF. In general, CF has not been included in drug development programs for this big group, but we're working to try to make sure that they are no longer excluded and that we can, in a smart way, pull them in because there's a lot of active drug development work happening in that space. I mentioned work that we're supporting really for the first time to set a goal of developing a new diagnostic tool or assay to detect lung infections without using sputum. Going to highlight a few ways we're doing this now. On the left are companies that we've partnered with and are working with who are developing projects like this and now working to develop assays specifically, at least initially, to detect Pseudomonas, but using that same technology as success is seen in bacteria beyond just Pseudomonas. So the top two, Pearl and MyDX, are developing different urine-based approaches to determine whether the bacteria is present not in the bladder or the urine, but really whether it's present in the lung and show some biomarkers or signals that can be seen in urine. And the bottom is a similar approach by a group called Alstone in the United Kingdom, working on breath that's collected and that can be sent to the lab to determine if there's certain signatures that tell us what infections are or are not present. On the right, these are academic or university-led research groups. So at the top is National Jewish Health out of Denver. They are developing a urine-based test for mycobacteria, specifically Bacterium abscessus. In the middle, University of Washington, where researchers are using blood and very sophisticated assays in labs to determine again what bacteria are in the lung and not in the blood. And so we're excited to see how that progresses. And then at the bottom is an important study that's really supporting all of these and even future companies or universities that we'd like to support and work with. That's a study being led by Gina Hong out of Penn. We would appreciate your help if you're at sites who end up doing the SENS-CF study, which is just getting going. So what we're doing here in brief is that when people are able to come to clinic and provide a sputum sample on that day, then we know pretty well what their bacteria may be based on a culture. And at that same time, we want to collect some other samples from them and bank and store those so we can support studies like this. So we may be asking people to provide some blood or breath or urine or other samples at that time. Let's shift then for a moment to quickly review some of the drug development activities that have and continue to happen at the foundation. This is drawn down from our website, and you can go there and review these in more detail. At the top, it just highlights, though, a reasonable amount of success that has been seen to develop antibiotics for people with CF. This is largely through the same kind of approach, which is to take antibiotics that are already approved and used by IV delivery and convert them to safe and more effective inhaled therapies for the chronic lung infections in people with CF. So that is great to see a number of options now available. But you'll see at the bottom, those that aren't as far along in development. This is where we're working now. And here we're taking a more non-traditional and hopefully more innovative approach, often using biological systems like phages, which you'll hear about in a few minutes from one of my colleagues, or antibodies that can disrupt the biofilms that those bacteria can hide in. So the immune system can clear up those infections or antibiotics can be more effective. I want to highlight a couple of these here. This AZD program. This is a program being led by AstraZeneca, a relatively large company, and they're developing an anti-pseudomonal agent for that large bronchiectasis community. But they are willing to include people with CF as part of that bigger umbrella. And so that's an example of that strategy that we're trying to support. And then Alstone at the bottom, as mentioned, is one of the diagnostic programs, specifically trying to see if breath might be a capable substitute for sputum. Lastly, before I turn it over, I want to just call out a few of the academic or university-led projects that fall nicely into five themes. This is by no means exhaustive of the academic research being done that we're excited about, but a few themes we wanted to share with you. First of all, we want to understand how best to use the antibiotics we have. And that includes people with some of these more challenging but perhaps less common infections, such as Achromobacter, Burkholderia, mycobacteria, for example. We also want to understand how best to treat exacerbations in the current era. Do we need all the antibiotics we've used in the past? I think is a key question that is on the minds of many care teams and also those with these infections. We've, for now, many years been focused on trying to best understand how the modulators, for those able to use them, are changing infections. And so there are a couple of large studies that you may be familiar with. Promise, which is closing now, and we're beginning to see many more papers come out reporting our findings, and then begin studying when these modulators are started at a pretty early age. Does that have a different impact as opposed to perhaps starting when you're in your teens or adulthood? As mentioned, we're supporting studies on how to detect bacterial infections in the lung without needing to use sputum. And so I mentioned the SENSE study. That's one of the big ones. Also, how to include people with CF in these large bronchiectasis drug development programs. And so on the one hand, want to make sure that people with CF can get access to that research and eventually those drugs if they prove to be safe and effective. On the other hand, we want to make sure that we understand that just being included doesn't hide from us whether or not the people with CF are truly benefiting and not just the large population. So we have to be somewhat thoughtful on how we take this new approach. And lastly, many of you are aware of mycobacterial infections as particularly difficult to treat. The treatment burden and side effects are substantial, as Sammi highlighted. And so a couple of programs focused on how to better treat, perhaps with innovative or different ways, some of these mycobacterial infections. And on that topic, I'll turn it over to my colleague, Stacy Martiniano, who will tell us more about mycobacterial disease.
Thank you, Dave.
Apologies, I'm trying to stop sharing.
That's all right. All right, looking good. Let me get mine now. All right. Well, thank you so much. Hello, everyone. Thank you for being here. So as Dave mentioned today, I'll provide an update on research really focused on nontuberculous mycobacteria, or NTM, and some other special pathogens in cystic fibrosis. I'll review what we know about NTM and discuss several ongoing research studies and share how these efforts are improving care and hopefully creating some new treatment opportunities for people living with CF. So I'm sure most of you know that NTM are bacteria commonly found in water and soil, and they can infect lungs of people living with CF. Over time, approximately 1 in 5 people with CF may have NTM detected in their sputum or other respiratory cultures. Our current studies aim to improve early diagnosis, understand who's at risk for disease progression, develop more personalized treatment approaches, and ultimately improve quality of life for people with CF and NTM infection. And importantly, these studies are multi-center collaborations, and they are actively involving patients and families, and they help guide evidence-based, patient-focused infection management strategies for NTM and other special infections. So one of the most important things we've learned about NTM and cystic fibrosis is that not everyone who has NTM detected will have the same health experience. So for some people, NTM may appear only once in a respiratory culture and then never be seen again. This is often referred to as a single or transient detection infection and may not cause symptoms or long-term health problems. For others, NTM may be found repeatedly over time, but without clear signs that it's harming their lung health. This is sometimes called a persistent or an indolent infection, and careful monitoring may be all that is needed. A smaller group of people who develop what is known as NTM lung disease have repeated detection of NTM, and it's linked to worsening symptoms or changes on lung imaging or worsened lung function decline. So research has shown that while about 1 in 5 people with CF may have NTM detected at some point, only a portion of these individuals develop lung disease that requires treatment. So understanding these different patterns really helps patients and care teams avoid unnecessary interventions, but certainly identify quickly those who need closer follow-up and treatment. And this approach really supports more individualized and thoughtful care. The PREDICT study is the first study I'd like to highlight. PREDICT was designed to try to help answer the key question, after NTM is detected, who will go on to develop NTM lung disease? This study follows people with CF over time to understand who develops NTM lung disease after detection, and the study aims to identify early warning signs, disease progression, and help distinguish between simple infection and lung disease that requires treatment. An important component of PREDICT is evaluating the impact of CFTR modulators and how they affect the risk of developing NTM lung disease. And this study supports early detection, patient-reported symptoms, confident treatment decisions, and future research opportunities. While PREDICT focuses on lung disease development, the patient study focuses on understanding how well treatment works. This study also follows people with CF, but those subset with NTM lung disease to help understand treatment tolerance and success. Patients aims to improve decision-making and guides therapy choices for better patient outcomes. We know that NTM treatment is very challenging and requires multiple antibiotics over long periods of time, as well as careful monitoring. So patients helps us understand which treatment strategies are most successful and lead to infection clearance, how well patients tolerate therapy, and what happens to lung health during and after treatment. So several important lessons have emerged from the PREDICT and patient studies. Related to NTM detection and monitoring, most patients with NTM don't require immediate treatment and can be managed with careful monitoring. We've seen that structured treatment approaches and prospective studies like patients are helping improve treatment success compared to the past. And we're seeing that personalized and follow-up care remain essential. So specifically, personalized care, really strong data collection, and long-term follow-up are critical to ensuring appropriate attention and treatment for each individual and to help inform for future care strategies, not only for individuals, but for all people living with CF. As I mentioned, we're monitoring how CFTR modulators are changing the landscape of NTM and CF. As Dave mentioned, we're seeing that CFTR modulators reduce sputum production, but we're also seeing that among those with NTM, we are seeing lower rates of NTM detection in respiratory samples. However, despite modulators, NTM has not disappeared. We have seen that some patients still develop new NTM infections despite being on modulator therapy, and this really highlights the need for continued monitoring. And understanding how modulator affects people in NTM infection through our research ideally will help clinicians adapt different screening approaches and follow plans in the clinical care setting for better patient care. One of the most exciting and innovative areas of treatment involves bacteriophage therapy. I'll just briefly introduce this topic and specifically talk about a phage study in NTM, and Becca will expand on this topic next. So phages are viruses that specifically target and kill bacteria, offering a novel approach to treat NTM infections. Phage therapy has been studied for patients with lung disease who do not respond well to standardized antibiotics. And phage therapy offers a hope as an alternative, especially when conventional treatments fail. And this gives us an opportunity to expand antibiotic research. So specifically the post-STAMP study assesses the safety and effectiveness of an NTM phage that is combined with existing antibiotic treatments. It's an added optional study linked to patients for people with CF and who have difficult to treat NTM lung disease, specifically those with Mycobacterium abscessus infections. So right now we are in the kind of ending phases and wrapping up our predict patients in post-STAMP NTM studies in the US. And the CF Foundation is using information from what we've learned in addition to data from the CF registry, and they are building a new national special infection research network that's called SPARC. Now, SPARC stands for the Special Pathogens Analysis and Research Consortium and plans to unite experts from multiple US CF centers to promote collaboration and accelerate research progress on infections. Right now, the focus is on rare infections. So the consortium aims to specifically understand and track disease progression of special pathogens in CF. This includes NTM, but we are also expanding to include Burkholderia and Achromobacter. SPARC will support data collection and research, as well as high quality and specialized laboratory services across the network. And ultimately, of course, the goal is to improve patient outcomes, and that we feel like by sharing expertise and resources, SPARC can enhance clinical care, evaluate new therapies, and innovate research and provide innovative research approaches nationwide. So I'll just end by summarizing why we think this research matters to patients and families. Infection research helps us make diagnoses earlier and more accurately, which can ideally reduce patient uncertainty and anxiety. Research can help identify who needs treatment and when, and this really promotes personalized care and ideally will minimize side effects. These studies help us explore new therapies, offering more options for difficult to treat infections. And most importantly, our progress depends on the participation of patients, families, and the CF community, and we thank you all for that. So with that, I'll hand this over to Dr. Rebekah Dedrick to present to us more on phage therapy.
Thanks, Stacy. Okay, hi everyone, and thanks so much for joining us today. Over the next 10 minutes, I want to give you an update on bacteriophage therapy in CF, We will talk about what phages are, what we've learned from the most recent studies, and how the foundation is continuing to invest in this promising area of research. So let's start with the basics. What are phages exactly? Bacteriophages, or phages for short, are tiny viruses that only infect bacteria. They don't infect our human cells, which is a really important point when we're thinking about safety. A helpful way to picture them is as highly specialized hunters. Antibiotics act more like a wide net. They can affect a lot of bacteria at once, including some good ones. Phages are much more selective. Each type of phage is essentially designed to attack one specific harmful bacterium, almost like a key that fits into a lock. That specificity is why researchers are so excited about phages. We're hoping that they can treat infections that are difficult or even impossible to treat with antibiotics alone. So why do we think we need something new? Why can't we just rely on antibiotics? Let's talk about that. For many people with CF, chronic lung infections are still one of the biggest day-to-day challenges, even now in the era of highly effective modulators like Trikafta. Pseudomonas aeruginosa is one of the most common bacteria we see in the CF lungs, and it is one of the hardest to clear. Antibiotics remain a critical tool. We're not walking away from antibiotics. But over time, these infections can become harder and harder to eliminate, and some bacteria become resistant. That's exactly why the Foundation has invested so heavily in anti-infective research, and why we've been pushing hard on phage therapy as one of the several promising new approaches. One of the most important recent chapters in that story is the BiomX trial. Let me set the stage. Before I talk about BiomX specifically, I want to give you a little context on how phage therapy has been used in CF up until now. Until recently, almost all of phage experience in CF came from very small studies in individual patient cases, sometimes called compassionate use cases. These are situations where a single person, often with a very difficult infection that had run out of options, received a custom phage prepared just for them. These cases have been incredibly valuable. Some of them have had really encouraging results, but they can only tell us so much. They involve one person at a time, and they're not designed to prove whether a therapy is broadly safe and effective for the wider CF community. For phage therapy to move from being an experimental, one patient at a time approach to something that could be approved and available for many people with CF, we need large, rigorous clinical trials. That's the milestone BiomX represented. BiomX developed an inhaled phage therapy called BX004. This targeted Pseudomonas. Earlier, smaller studies suggested that BX004 could reduce Pseudomonas bacteria and appeared safe over a short treatment period. Based on that, BiomX launched a phase 2b study, which became one of the largest phage therapy studies ever conducted in CF. So what exactly happened in that phase 2b trial? The bottom line is this: the phase 2b study was stopped because participants receiving the phage therapy experienced unexpectedly high rates of pulmonary side effects. This was the very first and only report of adverse side effects associated with phage therapy in humans. Patient safety always comes first. When those signals appeared, BiomX discontinued the study. The company later entered bankruptcy proceedings and as a separate business event, but it did affect what happened next with the program. And I want to be honest about the science. Researchers do not yet fully understand why those reactions occurred. Understanding that why is now one of the most important scientific questions in the field. This was, of course, disappointing news for everyone, patients, families, and researchers. But I want to reframe it in this way. Not every study succeeds, and every study teaches us something important. This trial revealed real questions that we now know must be answered before phage therapy can move forward. So what exactly did we learn, and what is the Foundation doing about it? A few really important things came out of the BiomX experience. First, it highlighted how important it is to understand how the immune system responds to phage therapy. That appears to be a big part of this story. Second, researchers gained valuable safety information that simply didn't exist before this trial was run. And third, these findings are already shaping the next generation of phage programs, from how phages are prepared to how patients are monitored. Here's how CFF is responding. We are preserving samples, study materials, and data from the BiomX program so that researchers can dig into what happened and learn from it. Follow-up studies are already underway to understand why those side effects occurred, with the goal of making future phage therapies both safer and more effective. I want to be clear on one point in particular. The Foundation did not walk away from phage research after BiomX. We're using what we learned to improve future approaches. Let me show you what those future approaches actually look like, starting with Sniper Biome and some of the academic research that we support. In 2025, the Cystic Fibrosis Foundation committed up to 6.5 million euros to support a brand new phage program with a company called Sniper Biome based in Denmark. What makes Sniper different is that they're not just isolating phages found in nature. They're engineering phages specifically for the challenging infections we see in CF. And critically, the whole program is being designed around bacteria collected from people with CF and grown under conditions that better mimic the CF lung environment. That's a direct application of one of the biggest lessons from BiomX. We have to test phages under conditions that actually reflect the lung and not just a standard lab dish. As of mid-2026, the program has successfully identified its initial panel of promising phages and is progressing through the planned development milestones. So the way I'd summarize this slide is, one of the most encouraging lessons from BiomX is that we need to understand phages better. And Sniper is applying those lessons from the very beginning. Now, companies like BiomX and Sniper are only part of this picture. So I want to highlight that there is a very active academic phage research community, and the foundation is supporting that too. There are two buckets into which phage research falls. The first is an investigational bucket, where researchers are really focusing on the basic science around phages, such as the discovery of new clinically useful phages and then characterizing them, understanding interactions between phages and the immune system, and developing methods to engineer phages. The second bucket is where researchers are bringing phage therapy to the clinic. This includes compassionate use phage therapy and academic-led trials like PostAMPs, which Stacy talked about earlier, and is a CFF-supported multi-site academic trial led out of National Jewish Health in Denver. PostAMPs is testing personalized phage therapy for nontuberculous mycobacteria, or NTM, lung infections in people with CF. NTM is a completely different kind of bug from Pseudomonas. It's notoriously difficult to treat, and current antibiotic regimens can take more than a year and are often poorly tolerated. Phage therapy is one of the most promising new options being explored in this area. So please take away this. Companies like Sniper and both investigational and translational academic programs are advancing in parallel, and CFF is actively supporting both tracks so that we can learn as fast as possible. Let me close with why, despite the setbacks, I've remained genuinely hopeful about phage therapy. Phage therapy overall remains one of the most exciting new approaches for new bacterial infections. We've learned important lessons from BiomX, and new programs like Sniper and the academic work in PostAMPs are applying those lessons as we move forward. The Foundation continues to invest in innovative infection research because people with CF deserve more treatment options. The story of phage therapy is still being written. There have been setbacks, real ones, and I don't want to minimize them, but each step brings us closer to understanding whether phages can become a safe and effective option for people living with CF. We haven't reached the finish line yet, but we are learning, we are improving, and we're continuing to invest in promising new approaches to fight chronic infections in CF. Thanks. Now, Sammi, if you'll join us and talk about some actions you can take on the last slide.
Thank you. Thank you to all of our wonderful panelists. You're going to hear from them in just a minute to answer your questions. But before we get to that, we want to tell you about 3 actions you can take. So you can tell Congress to co-sponsor the Pasture Act. The Pasture Act is a bill that is designed to combat antimicrobial resistance by incentivizing the development of urgently needed new antibiotics and antifungals. So you can urge your member of Congress to sign on as a co-sponsor. You can join Community Voice to share your input. I myself am a member of Community Voice, and they're pretty awesome. You will have opportunities to join focus groups, review and comment on updated infection prevention, and take the annual CF Insight survey to share what would make a real difference in your day-to-day life with CF right now. And you can find out more about currently enrolling phage trials by talking to your doctor and contact the clinical trial navigator, who is a design person who can provide one-on-one assistance. All of your website info is down below. So I want to now invite the rest of the panelists to come back up with me so we can get into the Q&A. Welcome, everyone. So the first question that I have is for both Becca and Dave. Are there any anti-infectives besides Colston, I hope I'm saying that right, that target biofilms?
I can start, Dave, if you want. Okay. So biofilms are essentially a large clump of bacteria that are attached together. Bacteria that are growing this way are often harder to clear from the lungs. So researchers are taking several approaches. These include phage therapy because phages can actually penetrate biofilms. And there are also anti-biofilm therapies that actually collapse the biofilms themselves. So the goal here is not only to kill the bacteria, but also make them less harmful and easier to clear from the lungs. Dave?
I think you covered it well. I would highlight one program for the person who sent the question may be very interested in this is Claramedics, is a company that the foundation is supporting, and they just finished a study in people with CF, and their drug specifically targets biofilms. And it looks like the early results, it's an early study focused on safety. That was positive, but there are some early findings that it may work. And so we're excited. It's a very novel kind of biology causing those biofilms to collapse and making the ability to kill those infections easier.
Thank you both. The next question is for Becca. It appears that staph has a small colony variant, which is more resistant to antibiotics and host immune responses. SCVs are considered more problematic and may cause greater damage to lung function. Can current CF center sputum cultures distinguish between regular staph and SCVs? If they can, are there any differences in treatment approaches?
Yeah, good question. So some CF microbiology labs can identify small colony variants, but they're not always detected on routine cultures because they grow differently than regular Staph aureus. SCVs may be associated more with persistent infections and greater antibiotic tolerance. If they are identified, clinicians may adjust treatment choices, but there aren't separate standard treatment guidelines specifically for SCVs.
Thank you. I have another question for you, Becca. Can you elaborate on what the side effects were in the BiomX study?
Yeah, so unfortunately, the patients who received the phage therapy um, ended up with pulmonary exacerbations. Those were the pulmonary adverse events that were associated with the drug product there. Patients stopped taking the drug product when those side effects occurred, and they recovered from those side effects. So as I spoke to in my presentation, the foundation has acquired all of the participant samples as well as the phages that were used in that study. And we have research ongoing to find out hopefully what caused those side effects and ways that we can prevent that in the future.
That's great. Thank you. I have one more question for you. Is specific phage going to have to be found for each patient with NTM?
Good question. So currently, for NTM infections, yes, that is the case. So typically, the patient's isolate would be cultured and tested against a panel of phages to look for phages that could infect and kill each patient's isolate because sometimes that varies. So as of right now, there isn't an off-the-shelf cocktail that you could use for NTM infections.
Thank you. So the next question I have is for Stacy. Can NTM bacteria be treated at home?
Yes, ideally that is what we aim for. So I will say that the majority of providers, and myself included, recommend that we start treatment in the hospital. These are typically new antibiotics that you may have not received before. And a lot of times we're needing to do that IV through something like a PICC line or a port. So we typically start treatment in the hospital, make sure that the drug levels are right, and we have a good monitoring approach that you're not having any initial side effects. But then the goal is to transition that you would take those medications at home. I also have this strategy, even if we're just doing just oral antibiotics. I think same rules apply. We want to make sure you have a good plan, that you're not having initial side effects. But then the goal is that you would go home and either self-administer these drugs or get some help from home nursing agencies and would do that treatment at home. We typically recommend checking in with your CF clinic monthly. Those maybe can be spaced over time, but this is a lot of antibiotics, a lot of monitoring, and we want to just make sure we're doing the right thing and that you're feeling the benefits from the treatment and that we're minimizing side effects.
Awesome. Thank you. Dave, this question's for you. What's the best theory as to why infections like Pseudomonas and MRSA are still showing up while on Trikafta?
Yeah, it's a good question. And I guess two parts. One is perhaps why most people who start Trikafta don't get rid of those infections. Why are they still there? And then secondly, are we seeing new infections identified in people who are on these modulator drugs? So the first one, why aren't they all cleared? If we're totally honest, we don't know all the reasons, and they probably are not, down to a single answer. But some of the theories, which I think was the question, is so bacteria, when they've had a chance to adapt and evolve in an airway over many years in particular, they can develop into a form of infection that doesn't typically occur in nature. Pseudomonas is around in the environment pretty broadly, but in the airway, they select to persist and grow in that airway. And so it can be much more difficult to clear that out than it was originally. Biofilms is part of that, but there are other factors involved. Secondly, though, probably just as importantly, bronchiectasis, that term I mentioned earlier, that damage that happens to an airway when it's chronically infected and inflamed, where it becomes somewhat larger and floppy and doesn't clear as well. So we know from that big group of people who have bronchiectasis and don't have CF that they actually have a Pseudomonas infection rate just like CF. 25% of that population grows Pseudomonas. And so just that by itself, even if you fix, so to speak, the CF issues that cause that initially, that bronchiectasis tends not to recover and go away at least completely. And so that's likely a major risk factor. Switching briefly to getting new infections, that also can occur, probably related to some of the same things. So the modulators, while remarkably effective for some, are not, as fully effective for everyone. And even when you get your sweat chloride down below a certain level that may be technically normal, it doesn't mean the airway is operating entirely normally as was suggested. So we are seeing, though at a much lower rate, some new infections and people who didn't seem to have these infections before, and then years into drug use, they have developed them.
Thank you. The next question I have is for Dave and Stacy. If you are unable to take a full Trikafta dose, can that increase your risk of picking up NTM?
Go ahead, Stacy.
I think this is certainly variable. We have seen people who have side effects with full dose of the modulators but have really nice clinical response on lower dose. I think the key is that you would take as much as you can tolerate and do that safely. And that every bit of CFTR restoration, we think, is helping to move those airway secretions to less sticky, able to help you have reduced inflammation in your airways, and all those things we think lead to your ability to not only reduce your risk of acquiring infections like NTM, but ideally are helping you be able to get rid of those NTM infection by improved mucociliary clearance.
Yeah, and I would just add that probably isn't restricted to NTM, probably other infections and other aspects of health. And it's also clear that not everyone metabolizes these drugs the same way. And so there are probably people who get their own max benefit at a lower dose. So there is some differences in what we call pharmacokinetics between people. And that's an active area study by researchers now. Yeah, but fully agree with Stacy. The maximum tolerated dose one can take safely is probably the best approach.
Thank you both. So the next question is for myself. It says, do you have any advice for managing the psychological impact before, during, and after an infection? I wish I had a super philosophical answer for this, but to be really honest with everyone, it was a really tough time for me. And I'm still really not on the after side, so I'm still kind of going through that. If I could say one big piece of advice about it, it would be to lean on your support system, whether that be your care team, your family, your friends, a therapist. And your mental health is just as important as your physical health. I know for myself and my experience with MAC and infection, I was constantly making decisions about my physical health and safety, but my mental health started to become not great during that time. So it was really important that I take care of my mental health. And as one got better, so did the other. More so, the more I started taking care of my mental health, I was better equipped to take care of my physical health. And there's no shame in taking care of your mental health or going to therapy or seeking those resources. So just lean on the people that you feel like you can during that time and know that there's a whole network of people with your team and the foundation that are standing behind you, and everybody wants to see you succeed and be here for you. So with them. The next question is for Stacy. Any data on why some patients get NTM and others do not?
Thank you for the last answer. It was excellent. So that is certainly an area of study that it's unique that our PREDICT study enrolls people who already have their NTM. So a lot of our focus has been on what are their characteristics and what is contributing to why someone might get disease, for example. But before that infection, we kind of lean on data from people with CF, but also without CF. We are looking to expand information that we can get from the One Bronchiectasis Network. We feel like there must be some combination of both the host, which means something in the person, inside their lungs, or something about their immune system, but also something about the bacteria. Certain bacteria live in certain environmental sources but they're everywhere. Why don't we all get this infection? So something about certain types of bacteria, if we can get clues from that, where they are in the environment, because then we can perhaps have more strategies on how to avoid them. I will say, kind of just generally speaking right now, that is a really strong focus of research. We don't have very clear guidance. But that being said, the CF Foundation is currently sponsoring new NTM guidelines for the CF community specifically. And this is an area of focus that we're going to look at the literature and provide some summary statements on. So that's something that we're working on this year.
Awesome. I have another question for you, Stacy. What are the typical treatments for MAC these days? A decade ago, it was azithromycin, inhaled amikacin, and ethambutol for more than 18 months. You mentioned IVs. Do they have some for NTM now?
Yes, Mac, we have more antibiotics in our toolbox, I will say. Um, we are still at our core using ethambutol, rifampin, and azithromycin. Rifampin does interact with the modulators, and so alternatives are being used. We reserve the IV treatment. I think Sammy mentioned this. For people with MAC, that are maybe categorized generally as more severe. So that could include having a very significant structural lung disease in your CT scan. You could have smear positive infection on your culture. You could have fever, a lot of systemic symptoms. And in that case, we're using IV forms of amikacin. That additionally, I just want to point out that there is one single FDA approved medication for NTM. Specifically, it's for MAC, and that is the liposomal version of inhaled amikacin. So a very specific inhaled amikacin as opposed to nebulizing the IV formulation. And those are for people with MAC that has been resistant to treatment. So a lot of similar antibiotics. At the same time, though, whenever there are new antibiotics that come out, whether they were designed to treat MAC or not, we do studies in the lab to see if they have any killing properties for MAC or abscessus or other NTM. And if they do, we add that to our toolkit of options for people to consider when we're treating these hard to treat infections.
Thank you. So the next question is for myself. It says, did Sammy clear the NTM? Yes, I think. So I had a lobectomy and I had the lowest lobe of my right lung completely removed because I had pretty serious encapsulated bit of MAC that had just been awful to try and get rid of. And then I went on to maintenance drugs to try to clear anything that was residual, one of those being a clinical trial that I am currently in for MAC treatment. So far, I've had one bronchoscopy, did not grow anything. I have another bronchoscopy next week, also hoping to not grow anything and be finally clear of MAC. So I hope so is the short answer to the question, but it seems like it. So the next question is kind of going out to everyone. Are CF patients at a higher risk with bacterial GI illnesses, especially with all the current outbreaks? For some, these bacterial GI illnesses it is recommended to only treat with antibiotics if it's severe or you are immunocompromised.
I guess I'll start. Not that I'll answer. It's a challenging question. It's definitely apparent that the GI tract of people with CF is often impaired or different than people without CF. Speaking mostly of the general healthy population. Um, we know that the microbiome or the complex ecology of bacteria that particularly protect the colon from some other bad infections is also altered in CF. So that may be a risk factor. The current outbreak, as I understand it, is a viral driven illness. And so I'm not personally aware of major susceptibility to viral GI illnesses in people with CF beyond the general population. We haven't seen that to my knowledge in general. That being said, usually hand washing is the best strategy and avoiding potential contaminated sources. And so that's good general advice for everyone. I don't know, Becca or Stacy, if you have any other information.
Do we feel like that covered it? We all satisfied with that? Awesome. Okay. Next question is for Stacy. Have you seen a difference in treatment success for NTM and Pseudomonas with patients who are pancreatic insufficient versus pancreatic Insufficient. I'm not sure what that was supposed to say.
Probably pancreatic sufficient versus insufficient. Um, and actually, we have not seen a difference quite yet in the people that are in our studies from a pancreatic sufficiency perspective. Thank you. I'm typing some more in the chat too.
Awesome. Thank you. So this is our last question going out to everybody. What are you excited about in the future of CF infection research?
I can start. I think phage therapy still remains one of the most exciting emerging approaches for difficult to treat bacterial infections. It's still experimental, and our responsibility is to generate the evidence needed to determine whether it's safe and effective for people with CF. But I think it will add another tool to our toolbox.
Stacy's typing, so I'll go. Thank you for a double duty. It's only been the last couple of years that the people at the foundation have felt like it was viable or reasonable goal to say we might be able to clear out or get rid of some of these chronic bacterial infections like chronic Pseudomonas, other ones, Burkholderia, etc. So uh, I'm excited to see what may be possible because science has improved. New approaches have come online. We're excited about both universities and companies that are pushing the boundaries there in ways that we think could be important based on the biology we have learned about these CF infections, and also based on some improved health that some people are experiencing through the modulators and less infection in those lungs. So in my opinion, that would just be really transformational to be able to clear these out, reduce the worries, maybe reduce the treatment burden, knowing that they might come back at some point. But I think that would still be an important step forward. So I'm excited about that.
Sammi, please tell me the question again as I was distracted. What I'm most excited for, perhaps?
Yes, what are you most excited about in the future of CF research?
Thank you. I'm really I mean, I think about this all the time and in my clinical in my clinic and in my research, and I'm just really thankful that to hear your voices that this is still a top worry because it's certainly something that I worry about, and I'm happy for the CF support of this. Just because infections are rare, these certain, especially these ones we've been talking about today, doesn't mean they're not important and impactful. And I think as opposed to just siloing and making us all choose our treatment path individually, pulling all that data to get more data on what works best, and ideally trying to reduce the length of treatment. Because I think that holds us back in some cases where I still do worry about young children, despite modulators, as some things in the comments still are at risk for infection. We celebrate that it's less, but we don't want to lose sight that it still could be important individually. So we want to have a good strategy. So I'm excited for getting some answers to these questions.
That's great. Thank you all for sharing that and being here and your presentations and answering everyone's questions. Thank you everyone for joining us for the webinar. You will be emailed a copy of the recording from tonight, and there is an email over in the chat box to send any questions that you have. And that is all we have tonight. So I hope everyone has a great night, a great rest of your week, and we will see you all very soon.